Hello, today we're going to be covering a pretty relevant topic considering the current pandemic as well as the recent news of a potential vaccine for the SARS-CoV-2 virus, aka COVID-19. And so today we're covering the immune system, and this is chapter 27 of your textbook. And hopefully this can provide some light on what's going to be happening with the vaccine and how your immune system responds to viruses and how it responds to not just viruses, but all other pathogenic bacteria, fungi, protists, etc., that somehow get into your body one way or another and try to take over the system. The immune system's job is to protect the body from various pathogens, and these pathogens can be bacteria, viruses, protists, fungi, and several species of parasitic worms. And so the body has three forms of defenses. First, we have physical barriers. These are our first line of defense, and they're almost like the walls around a city. They prevent the entry of the pathogen into the body in the first place. Because if a pathogen can't enter the body, it can't cause disease, right? And so some examples of this are your skin, tears, and mucous membranes. They're responsible for keeping the pathogens out or trapping them so they can be removed before they can get into your tissues. If the pathogen gets past the physical barriers and gets into your tissues, our second line of defense kicks in, and this is our innate immune response, or also called the non-specific immune response. And it's called this because these immune cells will attack any foreign material that gets into the body, no matter what it is. They see it, recognize it quickly, and work to clear those pathogens as quickly as possible. And this section of your immune system is functional at birth. So once you're born, you already have this immune defense, and this is to help protect you just in case you get injured early in life, right? You don't have time to wait for your immune system to develop like some other elements of your body. You need to be protected immediately. So this is present as soon as you're born. And some cells that are involved in the innate immune response include macrophages, neutrophils, dendritic cells, and natural killer cells. And we'll talk about each of those cell types a little bit later. And then if the pathogen gets past the innate immune system, we have our third and final line of defense, and this is the adaptive immune system, aka the specific immune response. And this is called specific because these particular cells and this part of the immune system responds to specific pathogens. They don't just attack anything and everything. They attack very specific cells, very specific pathogens in a very aggressive manner, right? And so they have to do this because if the pathogen gets past the adaptive immune system, it can run rampant throughout the body and can cause you serious illness or death. This response is actually much slower than what we see in the innate immune response. So because in the innate immune response, they respond to any pathogen that makes it past the skin and mucous membranes, they can respond very quickly. But in the adaptive immune response, they respond to very, very specific pathogens and specific parts of pathogens. So it takes them a while to build up enough numbers of soldiers, if you will, the soldier cells, to fight the pathogen. So instead of with the innate immune response, if you get a cut, you are like almost instantly start to see it swelling and you feel heat and pain. That's your immune system responding almost instantly to that injury. With your adaptive immune system, you have to wait a couple of days for your body to actually mount a defense, and it may take a couple of weeks for your body to actually clear the pathogen. So it's a little bit slower process. But one amazing thing about this part of your immune system is it's also responsible for remembering any pathogen it encounters, so that if you get reinfected again at some point, your body is able to more quickly respond and clear that pathogen out faster than when you first encountered that pathogen. And the cells that are part of the adaptive immune system include your B cells and your T cells. Your integumentary system is your body's first line of defense, and it is composed of your skin, mucous membranes, sweat glands, oil glands, hair, and nails. All of these structures work together to protect your body from various stresses, injury, and pathogens, both internally and externally. And so the integumentary system protects the body by acting as a physical barrier to entry for pathogens, but also works to physically remove pathogens from the body. And so one example of a physical barrier is unbroken skin. As long as your skin is intact and is not broken, it is a very, very good protector. Pathogens have a very hard time getting past the skin, the vast majority of pathogens. And so as long as your skin is unbroken, then even if you do come in contact with a pathogen, it can't get into your body. But however, if your skin does get broken, even if it's something as small as a very small pinprick, that is a potential area of entry into the tissues. So you always want to make sure that you're covering up any scratches or abrasions that your body has, because you want to make sure you keep those pathogens out of your sensitive tissues. And then another example, when we're looking at the physical removal of pathogens from the body, we can look at the ciliated cells of the respiratory tract. So I've included a picture here that shows you what these ciliated cells look like. So when we follow this line, this is kind of like the cell membrane. Anything above this line, all this kind of fuzzy material here, are the cilia of the cells in your respiratory tract. And so when you inhale, you potentially expose your lungs to various pathogens that are just floating in the air, as well as when you talk. And so your respiratory tract has mucus that coats all of its surfaces, that's responsible for trapping any potential pathogens so that they cannot get into the tissues. Well, this mucus cannot stay in your lungs, in your respiratory tract, because that can cause major problems. As the mucus builds up and as the number of pathogens builds up, some of them may start to be able to survive in that area. So what your body needs to do is remove that mucus from the respiratory tract. And to do this, they use these ciliated cells. And so the cilia on these cells brush the mucus up from your respiratory tract to the back of your throat. And then once it reaches the back of your throat, you'll swallow that mucus down into your digestive tract. This is oftentimes where you see people like clear their throat, or they'll cough, or you feel that kind of mucus in the back of your throat and it makes you go that is your body's way of moving the mucus from your respiratory tract down into your digestive tract. And then once that mucus gets to your stomach acid, it will degrade any of the pathogens that had gotten into your respiratory tract. Another example of physical removal is tears. Tears help to lubricate your eye, but they also help to sweep away any pathogens that may try to infect the tissues of your eyes. So your integumentary system can use several chemical traps to kill, inhibit the growth of various pathogens. And so some examples I've included here are like enzymes. So there are enzymes in your saliva and in your sweat glands called lysozymes. And lysozymes help to break down bacteria by breaking down the bacterial cell wall. So this is one way of killing any potential bacterial pathogens that may get in and on your body. And then we have acids. So we just talked about the acids in your stomach are able to break down pathogens that are maybe in your food or get into your respiratory tract that get trapped in the mucus. But also the female vagina is also very acidic. And these acidic conditions are not conducive to the growth or survival of various pathogens. So these are another effective way of protecting yourself from pathogenic microbes. Then we have antibacterial and antiviral compounds that are produced in various parts of our bodies, including in our tears. And so these compounds are specially made by our cells to kill specific pathogens. And then finally, we have traps. These are generally some sort of mucus or thick secretion that is able to trap the pathogen so it can be removed from the body. So we just talked about the mucus of your respiratory tract. We also have mucus in our digestive system and ciliated cells in our digestive system that helps to sweep not only food and stool throughout the system, but also sweeps any pathogenic bacteria out of the system that it can be removed in the feces. We also have like your earwax is another form of almost like a mucus-like secretion where pathogens can get stuck. And then once they're stuck in that earwax, they can't infect your tissues. Instead, when you clean out your ears, you remove those potential pathogens and you're all good. So these are just a couple of major ways that your integumentary system acts as the first line of defense to prevent from pathogens invading your body. If a pathogen makes it past your physical barriers, it's now the job of your innate immune system to kick in so that it can identify and kill those pathogens before they spread throughout your tissues. And so the innate immune system is composed of a variety of different types of cells that all work together to identify and destroy various pathogens. And these immune cells are produced in the bone marrow, and then they're put into the bloodstream. And from here, they have two different paths. They can either remain circulating in the bloodstream like we see in neutrophils, or they can be deposited into various tissues around your body. And when they get to their tissue location, they will patrol the areas around the tissue to make sure that they don't see any potential pathogens that have gotten into the body or that are hiding in your tissues. And some examples of cells that are residing in the tissues and patrolling the tissues are like macrophages, dendritic cells, and natural killer cells. And we'll talk about all those different cell types in more detail in the next slide. And so the job of all of these cells is to recognize self versus non-self cells. And so what this means is all of our cells in our body produce lipids, carbohydrates, and proteins as just a natural part of a healthy cell's life. But many of these proteins, lipids, and carbohydrates are different from the proteins, lipids, and carbohydrates that are on non-self or pathogens and other microbes. And so what the immune cells do is they have various receptors that are on their cell surface. And so I've included this picture here to show you that there's just a variety of different type of proteins on the cell surface. So this is not an actual picture of a real cell. This is artistic rendering, but this is kind of what they're like, right? So they're covered in all of these receptors, and these cells patrol the tissues and patrol the bloodstream, and they're using these receptors to bind to various proteins in the tissues. And so as they're binding, if any of these receptors bind to something that's non-self, they then go into attack mode, and because they recognize that whatever they're bound to, it's not part of the body and it needs to be destroyed. And the innate immune system is called the nonspecific response because these proteins bind to a variety of shared substances. So I include this picture down here to show you that there are various types of receptors. These are actually the real names of the receptors. You do not have to memorize these. You do not have to memorize what they bind to. But it's just to show you that there are several different types of receptors, and each receptor binds to these compounds that are shared amongst a variety of pathogens. So if we're looking at one example, LPS. LPS is present in a variety of bacteria. So if it sees LPS, it knows this is not self. I don't know what it is, but it's not one of us, and we need to kill it. Same thing with peptidoglycan and lipoproteins from bacteria. These are common shared compounds that various species of bacteria will have, but the macrophages and dendritic cells and things like that, they don't need to know specifically what that pathogen is. They just know that it's not self, and we have to attack it and remove it. And so that's why it's a non-specific response. And so once it identifies that pathogen, it will usually undergo phagocytosis to kill that pathogen, and as well as any infected cells. So if a cell has a virus or bacteria living within it, they can also identify those infected cells and destroy them in a very similar way. And these immune cells also work to recruit other immune cells to come help with the fighting the infection. And so we'll talk about the chemicals called cytokines that are responsible for this recruitment in a couple more slides. There are several different cell types that make up the innate immune system. We'll talk about 5 now, and then I'll mention 2 a little bit later. So of the main 5, we have neutrophils, and neutrophils make up about 50 to 70% of all circulating immune cells. So these cells are made in the bone marrow, and then they're put into the bloodstream, and they, for the most part, stay circulating within the bloodstream, patrolling for any pathogens that may make it into the blood, as well as they just keep circulating until they're recruited to go to specific tissues to help fight an infection. They're very, very short-lived. They only live about 3 to 5 days, but their main job is to phagocytose whatever pathogens they find, and then after they phagocytose enough pathogens, they basically self-destruct. So they bring in the pathogen, and then it's almost like they explode, if you will, like they sacrifice themselves in order to kill the bacteria or virus or fungus or whatever it is. They also end up killing themselves. And there are other cells that come by and clean up that debris later. And they also can produce different chemical compounds to help them with fighting infections. And these include hydrogen peroxide and hypochlorous acid. And hypochlorous acid is very similar to the compounds you find in bleach. And so what they'll do is they'll put out these compounds into the tissues around them while they're ingesting various bacteria, viruses, or whatever the pathogen is that helps to kill the cells before they ingest them, and also to help slow them down, so it makes it a little bit easier to catch those pathogens. And because of that kind of self-destructive nature, they make a pretty big mess in the tissues where they're at. They're a big help because they're really good at killing pathogens. However, it is a bit messy, and that mess we can see as pus. So when you have a wound and you see that kind of that white, yellowy, or even greenish type of liquid that will come out, that is generally dead neutrophils as well as dead other cells, and both live and dead versions of the pathogen. So you want to make sure that you're not just touching pus, because there could be some live pathogen in there, but that's primarily what it is. It's just a response of your body fighting really, really hard to clear whatever pathogen has gotten into your body. Then we have macrophages, and macrophages are very large phagocytic cells that reside in the tissues. So these ones, they go from the bone marrow, circulate through the bloodstream until they get to particular tissues, and then they stay in those tissues and patrol that area just in case there's a breach. And they're really good at also clearing away pathogens, but also at cleaning up that debris from neutrophils when they basically just sacrifice themselves for the greater good, right? These cells can also produce hydrogen peroxide and hypochlorous acid to help with combating any of the pathogens that are in the tissues in their near environment. And they are also antigen presenting cells. So how this works is an antigen presenting cell is able to ingest a pathogen and then display parts of that pathogen on the outside of its cell membrane. And this is important because it then goes to your adaptive immune system and says, hey, we found this thing. And it shows your adaptive immune cells so that part of your immune system can kick in. And so how the whole process works from ingesting a pathogen to displaying it is shown really well in this picture here. So we start with the pathogen getting phagocytosed, and then when the pathogen is brought into the cell via a vesicle, the vesicle will fuse with a lysosome, which is this part of the image here. And that lysosome will start to break down that pathogen, which is what it's showing you in part 3. And then from there, once the bacteria or virus or fungus or whatever it is broken down, there's 2 fates to the different digested products. One, they can be removed from the body, right? Because they're now harmless. Nothing's going to happen. The pathogen has been broken down. Or the cell can load parts of that pathogen, so small pieces of protein or sugars or various different lipids, onto proteins, their own proteins, and then put those proteins on the outside of their membrane. And then from there, they'll go to the adaptive immune system and show those pieces of the pathogen to the adaptive immune cells to help get them started to be prepared just in case the pathogen makes it past the innate immune system. And then from here, we also have dendritic cells. And dendritic cells are also phagocytic cells that reside in the skin and in your mucous membranes. They're also presenting cells, and their main job is to catch a pathogen and just report to the adaptive immune system. They don't do a whole bunch of fighting the pathogen at the tissues. Their biggest thing is just to alert the adaptive immune system as fast as possible. So they'll present that pathogen, pieces of that pathogen on their membrane, travel to the lymph nodes and spleen, and then signal the adaptive immune system to kick in. And then we have our natural killer cells, and these are also called NK cells. And these are our antiviral cells. They also hang around in the tissues, patrolling the area, and if they find out that a cell has been infected by a virus, they poke holes in that viral infected cell and kill that cell. And when they kill that cell, it also kills the virus. So they're kind of like the soldiers that make sure that everybody's doing what they're supposed to do, right? So if the cell is infected with a virus and says, "Hey, I'm infected," those natural killer cells come by, poke holes in that cell, and then your body will go ahead and clear any of that cell debris and dead viral material and remove it from your body. And they can also recognize and kill cancer cells in a very similar way. And so these are our first kind of big four. In addition to the innate immune cells, there are several other substances that play a role in the proper functioning of the innate immune system. And these substances are so important that the innate immune system would not be able to function properly without them. One of these substances are cytokines. And cytokines are compounds that are produced by one cell that are released into the environment, whether it be the tissues around that cell or into the bloodstream, that then travel and interact with another cell to cause some sort of response. And the innate immune system uses these primarily for recruiting other immune cells to a site of infection. They can also use these cytokines to activate other immune cells to come and clear a pathogen more effectively. And so this picture here, I think, does a really good job of walking you through what cytokines do. So we start with a cell. So we say this is a macrophage cell for this example. And then there's some sort of inducing stimulus. In this case, if it was a macrophage cell, let's say that this cell, it engulfs or phagocytoses a bacterial cell. That would be its inducing stimulus. You don't have to remember this term, just remember that something happens that causes that first cell to produce cytokines. And so in this case, we're using a macrophage as an example. The macrophage will produce these chemical signals called cytokines and release them into the tissues and into the bloodstream. And those cytokines will travel throughout the body until they meet a target cell. And this target cell can be either another immune cell, or they can be a body cell in your body that does other things that aren't involved in the immune response. And those cytokines will bind to receptors on the membrane of the target cell, and they will cause some sort of response in those cells. So if this target cell was a neutrophil that's circulating through the bloodstream, that will target that neutrophil and say, hey, follow this signal, follow the cytokines to the area of infection. And that can cause them to do one thing. If they're cells that are in the blood vessels, it can tell those cells, hey, you need to spread out, become more porous, because we need to bring in substances to the site of infection. And if some of these cytokines go and travel through the bloodstream and bind to cells in your hypothalamus, they can cause you to develop a fever. And we'll talk about this process in a lot more detail in a couple more slides. But there are some cytokines that are responsible for you developing a fever when you have an infection. And then another type of substance we have are called interferons. And interferons are similar to cytokines, but instead of being produced by immune cells and having a target of other immune cells or cells in other cells in the body, instead interferons are produced by your cells that are virally infected. And then they produce these signals so that it warns all the cells around them that, hey, I'm infected, you need to protect yourself. So to give you guys an example, let's say we have a whole bunch of cells in the body in a particular tissue. And this cell here gets infected with a virus. Well, that cell will start to put parts of that virus on the outside of its membrane, and that will target the natural killer cells to come and kill those cells. It'll also, while it's producing those signals to the natural killer cells that says, hey, I'm infected, come kill me, they're also producing chemical signals that they release to the environment around them. And these interferons will tell the cells that are around that infected cell that, hey, I've gotten infected with a virus. You need to start producing antiviral proteins so that you don't get infected too. And so all those cells around that virally infected cell will have a heightened sense of security because they'll now have made these antiviral proteins that will help protect them from getting infected. So this is what interferons do. And then finally, we have our complement proteins. And complement proteins are a variety of different types of proteins that circulate through your blood system. And they play a role in protecting the body from pathogens because they're recruited to a site of infection and they can help to actually kill pathogens. And so they can do this in a couple of ways. One, these proteins can aggregate together and they'll poke holes in the membranes of especially bacteria. And when you poke holes in the membrane of bacteria, it causes water to rush into the cell. So that's what they're showing you in this picture here. All of these purple pieces here are the proteins, the complement proteins that have created a hole or a pore in the membrane of this bacterial cell. And that will cause water from the tissues in the interstitial fluid to rush into that cell. Well, when that happens, as we've seen with blood cells and diffusion and osmosis, that when water rushes into a cell, that will cause that cell to rupture and burst. And so that's what's happening. As more and more water enters into the cell will increase in pressure until it can't hold anymore, and it will burst. And that ultimately kills the cells. Complement proteins can also help to kill pathogens by sticking to pathogens and sticking various pathogens together. So they can act almost like glue. So they'll bind to this bacteria and stick it to this bacteria and stick it to this bacteria and this bacteria. And this helps the immune system because it's always easier to see a larger target than a smaller target. And so if you have a whole bunch of bacteria or viruses or something that's all been stuck together, then the phagocytes, the macrophages or dendritic cells, can come by and more readily and more easily identify those pathogens and engulf them and just kill a whole bunch of those pathogens at one time. And so these complement proteins are very important with helping the innate immune system to identify and kill these potential pathogens. One key indicator that the innate immune system is doing its job is inflammation. And inflammation begins when your physical barriers are breached. And so let's say you're sitting and you're reading a book and you flip a page and get a paper cut. Now that your physical barrier, which is your skin, has been breached, any microorganisms that were on the skin or on that paper have now been introduced into your body. And whether those microorganisms are pathogenic or not, we still don't want them hanging around in our body. So your body works very hard to clear those microorganisms as fast as possible. So just in case they are pathogens, they don't spread. So macrophages in the tissues near that site of injury will recognize the microorganisms as non-self and start to break them down through phagocytosis. While this is happening, the macrophages are also producing cytokines, and those cytokines will diffuse into the tissues around the site of injury and also diffuse into the bloodstream. And in both cases, they're responsible for recruiting more cells and complement proteins to the area of injury to clear out all those microorganisms that made it in when you got cut. Part of this recruitment effort is through activating two other innate immune cell types. These are basophils and mast cells. And basophils circulate through the bloodstream, similar to how we saw with neutrophils. And mast cells, they reside within the various tissues around your body. Both of these cell types are responsible for producing a compound called histamine. And histamine is important to the immune response because it dilates the blood vessels and causes them to become more porous. And this is important because if the immune cells are stuck in the blood vessels that are circulating, like neutrophils, and they can't get out, then they can't help, right? But when these blood vessels dilate, it allows the neutrophils to leave the blood vessels and come into the tissues to help with the fighting the microorganisms. It also allows for complement cells to leave the blood vessels and go to the site of infection as well. And then this dilation also helps with increasing the blood flow. And the increased blood flow allows for more immune cells to pass by this area of infection so that you can have more immune cells respond to that area. And so all of this, this increased blood flow and dilation of your blood vessels is actually the reason why you feel so much pain and swelling and redness and heat when you get injured. And that's because the redness is associated with the fact that there's increased blood flow. And then the swelling and the pain is due to the increased pressure in those tissues. So because the blood vessels are dilated, more fluid is leaving those vessels and getting into the tissues. When this happens, that causes the tissues to swell, and that swelling causes increased pressure, which you can feel as pain. And this is also why you feel heat in those areas. So it may be kind of like alarming, but all of those symptoms that you normally associate when you get injured are good. That's letting you know that your immune system is working. So once the neutrophils get to, and the complement proteins, get to the site of infection and they leave the blood vessels, they will also stimulate the mast cells and basophils to produce even more histamine to help keep the blood vessels open and to recruit more cells. And then also all of these cells, the neutrophils, the mast cells, the basophils, and the macrophages, throughout this whole process are continually producing cytokines to keep that immune response going until the potential pathogen, and even non-pathogen microorganisms have all been cleared. And so once all of the microorganisms have been cleared from the site of infection, then everything goes back to normal, right? All the immune cells stop producing cytokines, they stop producing histamines, the blood vessels will no longer dilate, all the neutrophils that didn't self-sacrifice will go back to the bloodstream to circulate, and everything goes back to how it was. And then if you get injured again, then this whole process starts over. I mentioned before that some cytokines are used to induce a fever. And so these cytokines are produced by macrophages in the tissues, and they travel to the bloodstream. And then from the bloodstream, they're sent to the hypothalamus, which is a part of your brain that's responsible for regulating what your body temperature should be. And once those cytokines bind to cells of your hypothalamus, it tells your hypothalamus to increase your expected body temperature. And that increase in your body temperature is what results as a fever. And so when you have a fever, you oftentimes will feel cold, and then you'll start to shiver. And that response is to help your body even further warm up. So you're not actually cold, like your body's not physically cold, but if your brain makes you think that you're cold, you'll start to shiver. And when you're shivering, you're making more heat, right? Because you're moving. And as you're making more heat, your body temperature will continue to increase and increase and increase, right? And so as this occurs, the increased body temperature helps to actually fight infections, because a lot of microorganisms, they are very heat sensitive, especially bacteria and viruses. If a temperature gets above a particular threshold, those microorganisms, particularly bacteria and viruses, will no longer be able to replicate, and then their proteins will start to degrade and they'll just die. So this is a way for your body to help the immune system fight off the infection. Once the pathogen has been cleared, the cytokines will no longer be produced, and your hypothalamus will go back to registering your temperature and within a healthy range that it should be. And when this occurs, you'll start to feel hot, right? Because your body temperature is elevated still, but your hypothalamus now says, oh, your body temperature needs to be down here. And so you start to feel hot and you're starting to sweat. And of course, we know from what we talked about before, sweating is a method of trying to cool your body temperature. And so you'll start to sweat, you'll start to cool down, and then everything will return to normal. Now, if this doesn't happen, if you either maintain a fever for a prolonged period of time, or if you get an excessively high fever, you should always go to the hospital. An excessively high fever does help to kill the pathogen, but it also helps to kill your cells, right? So if the temperature of your body gets too high, your own cells start to denature, and this can cause serious problems for you, right? And it can even result in death. And so this is one reason why a lot of parents get very, very worried when their children have a high fever, or if they have a fever for a prolonged period of time, because that can be potentially life-threatening. And if you end up going to the hospital and you have an extremely high fever, they'll give you anti-inflammatory medicines. And this helps to block the cytokines from binding to the receptors in your hypothalamus. And so it won't trigger your hypothalamus to keep you to shiver and raise your body temperature. Instead, it'll combat that to bring your body temperature back down to normal. If that also doesn't work, then sometimes they'll cover you in ice or various other things to cool your core body temperature down so that you don't start to undergo serious brain damage and organ failure. If a pathogen makes it past both your physical barriers and your innate immune system, we only have one more line of defense, and that's our adaptive immune system. The adaptive immune system is composed of two cell types, B cells and T cells, and both these cells are produced in the bone marrow, but they mature in different areas of the body. B cells mature in the bone, which is where they get their name from, B cells from bone, and T cells mature in the thymus, and that's where they get their T from. And both B cells and T cells make up a significant portion of the lymphatic system. Approximately 99% of the lymphatic system cells are either B cells or T cells. And these B cells and T cells have a couple of options. They can accumulate in your lymph nodes, which are around your body, or in your spleen, or they can just kind of circulate through the lymphatic system and in your blood, patrolling for any pathogens that might come around. Your adaptive immune system is responsible for two things. One, recognizing specific pathogens and clearing them, and two, remembering those pathogens so that if you get re-exposed, you don't get sick nearly as bad the next time. And this memory is what we refer to as immunity. And immunity is long-term protection against specific pathogens. And this is so that when you get exposed that first time, you might get sick. And it might take a long time for you to get better. But that second time, if you get re-exposed, this time your immune system is able to fight that pathogen off a lot faster than the first time. And so you don't feel nearly as sick and you get better faster. And this is where vaccines come into play. Vaccines are designed so that when you get exposed to the actual pathogen, you don't get sick or you don't feel nearly as bad as if you would have gotten exposed to the actual pathogen for the first time. So what vaccines do is once you get a vaccine, it stimulates your immune system because your immune system thinks that that's the actual pathogen, even though it's not. So this is why sometimes when you get vaccinated, you may feel a little sick. And that's because your immune system is responding to the vaccine as if it was the actual pathogen. This is a good thing because now once the immune system clears the vaccine, you get those memory cells. And then when you actually get exposed to the pathogen in real life, like the actual pathogen, your immune system says, ooh, we know what this is, and they fight it off a lot faster. So you may not even realize you were exposed because your immune system cleared it so quickly, or you may get sick, but your sickness is not nearly as bad as if that was your first time being exposed. So taking a look at this chart, I think it does really, really good at explaining what this process is like. So let's say you get exposed to chickenpox for the first time, or let's say you get exposed to chickenpox through a vaccine for the first time. Your immune response is pretty slow, right? The adaptive immune system takes a while to ramp up. It's not nearly as fast as the innate immune system. So it might take several days for you to ramp up enough immune response to actually clear the infection. And then once you start winning the battle and clearing the infection, the amount of your immune response will go back down, right? Because you don't need to be constantly stimulating your immune system. And so during this kind of going back to normal is where we will establish those memory cells. So that next time, let's say you're hanging around with your nephew or something, and he actually has chickenpox, right? So you've already been vaccinated, so you have those memory cells. So if you get exposed to your nephew who has chickenpox, this time it's a shorter amount of time for your immune system to respond very aggressively to that pathogen, and it will clear it a lot faster because your immune system is responding far more, your immune system response is far stronger this time, and then you can clear that pathogen out much quicker. You might not even notice that you were even exposed because your immune system just cleared that sucker out before you even had a chance to get sick. T cells and B cells recognize specific antigens. This is why we call it the specific immune response. They don't just recognize everything. And antigens are any molecule that can induce an immune response. So this can be various pathogen molecules like proteins, lipids, and carbohydrates. And so I put this image here to show you that any of these lipids here, if they don't look like self-lipids like we make in our bodies, they're potential areas to target, and as well as these sugars here and proteins, various proteins, anything that's not self is a potential target. And then these every pathogen, has multiple antigens. So like I was just saying, any of those things can be an antigen, but not all antigens are from pathogens. So we know that people who have allergies are responding to maybe like pollen or different compounds that come from different foods. Those are antigens, even though they don't come from pathogens. So anything that just stimulates the immune system is an antigen. So there are many antigens per pathogen, but each a B cell or T cell only can recognize one antigen. So if a bacterial cell has several different proteins on it, so we got these type of proteins, and let's say we got these proteins, they're not really all that uniform, and then let's say we got these sugars. All right, it's various antigens that, and then also you have the DNA of this organism as well, or whatever, or RNA or whatever, that can be an antigen. But each immune cell can only respond to one of those antigens. So we have an immune cell, another, and another, right? This immune cell only responds to the red pathogen. The red antigen. It can only see the red one. And then this immune cell can only see the green one. Like very crude drawing. And then the last one can only see the orange one. So just remember that. Various pathogens can have a lot of different antigens, but each immune cell only makes one receptor for a particular antigen. There are two types of adaptive immunity, humoral and cell-mediated. Humoral immunity is often referred to as antibody-mediated immunity, and it's responsible for clearing pathogens that are in your body fluids. So any pathogens that are circulating in your bloodstream or in your lymphatic system are cleared by this form of immunity. And the B cells are primarily responsible for this form of immunity, in addition to the proteins that they make called antibodies. And then we have our cell-mediated immunity, which clears pathogens that are residing within your cells. And T cells are responsible for this form of immunity. Both humoral and cell-mediated immunity are made possible by clonal selection. And clonal selection is the rapid division of activated B and T cells to clear the pathogen and remember that pathogen. So what happens is your lymphocytes, B and T cells, will recognize antigens on pathogens or antigens that are presented to them. So these antigens can be proteins, lipids, or carbohydrates. And let's say for this example, it's LPS. And LPS is a compound that's found in bacteria. So your lymphocyte recognizes that antigen using antigen receptors on their cell membrane. And that will cause that lymphocyte to undergo rapid cell division. And when they divide, they'll form two types of cells. The one group of cells become your effector cells. And these effector cells go on to actively fight the infection. So they're the ones that are going through attacking the pathogen and destroying them. And then the second group of cells are your memory cells. And these memory cells will circulate throughout the blood after the infection has been cleared and just patrol to make sure that you don't have reinfection. And so that these cells are the ones that are responsible for that enhanced response if you do get re-exposed. And clonal selection, this entire process, it occurs every time you get exposed to the pathogen. So if you get exposed the first time, second time, third time, 15th time, this clonal selection occurs. But the clonal selection process is a lot faster due to the memory cells upon second exposure than it was on your primary exposure. Looking more closely at the adaptive immune cells, let's start with the B cells. And B cells, as I mentioned before, are produced in the bone marrow and they mature in the bone marrow. And upon clonal selection, there are two groups of B cells that we get. One are our plasma cells. And the plasma cells are the effector cells. They're the ones actively going out there and helping to clear the infection. And they can do this through the use of antibodies. So we'll talk about antibodies in a lot more detail in the next slide, but for B cells, antibodies can perform two functions. If the antibodies remain attached to the plasma membrane of the B cells, then they act as the B cell receptor. And so as the B cells traveling around, they will encounter various antigens, and once that B cell receptor, which is a form of antibody, binds to the antigen that that B cell is specific for, that will allow that B cell to phagocytose the pathogen that the antigen is associated with and destroy that pathogen. Then B cells can also secrete antibodies into the bloodstream, which will then go and circulate around and bind to various pathogens that express that particular antigen and help with clearing the infection in several ways, which we'll talk about in the next slide. And then we have the second group of B cells that are a result of clonal selection, and these are our memory B cells. And these are the B cells that are going to be circulating through your bloodstream and through your lymphatic system, just patrolling to make sure that they don't see that antigen again, just in case you do get re-exposed. And B cells can act as antigen-presenting cells, similar to what we saw with dendritic cells and with macrophages. So how this works is basically the same way that we talked about with the macrophages and dendritic cells. So the B cells will use their B cell receptor, which is a form of antibody, to bind to their specific antigen. And then once they bind that antigen, they'll bring that pathogen that that antigen is attached to into their body, and then they will, well, not their body, into the cell, sorry, and then they will load that antigen onto a specific protein that's for exposing to other adaptive immune cells. So you don't have to remember what the name of these receptors are or anything like that. I just wanted to show you here that that antigen has been loaded onto this protein here. And then that B cell is like now saying, hey, I found this antigen, and it will interact with another adaptive immune cell. In this case, this is a T cell, and it will stimulate that T cell to have some sort of response. In this case, it's stimulating that T cell to produce cytokines that will make that B cell mature and go on to help fight the infection and undergo clonal selection. So they can do very, very similar functions as we saw in macrophages and in dendritic cells. The power of B cells lies in antibodies, and antibodies are composed of 4 polypeptide chains. We have 2 heavy chains, which are shown as this gold color in this picture, and then we have 2 light chains, which are exhibited by this orange color. And all 4 of these polypeptide chains come together to form this Y structure. And this Y structure is conserved amongst all antibodies. So all antibodies have that same Y structure. And antibodies have 2 main regions. You have your constant region, which is almost like the stalk of the Y. This constant region doesn't have that much variability. There are only about 5 different types in humans. And then we have the variable region. Variable. And the variable region is actually where the antigen binds. Each antibody can only bind to one type of antigen. So in order for your body to be able to recognize the various types of antigens that are possibly out there, we have to produce tens of thousands, hundreds of thousands of antibody types so that they can be recognizing various types of antigens. And so this picture up here is just showing you that if you want to bind this antigen here, this antigen, then you need to have a antibody that has a variable region that will bind it. This antibody will not bind to any other type of antigen. And similar thing here. If this antigen needs to have an antibody made for it, then this antibody is specific for that antigen and will not bind to any other type. So antibodies play a major role in helping to fight infection in a couple of different ways. One, they can help phagocytes identify pathogens and foreign material and infected cells. And they can do this in two major ways. One, they can bind to the pathogen or infected cell or foreign material. They can be like a venom or a piece of pollen grain or something. And then when they bind, it helps to signal phagocytic cells to come and engulf that pathogen or pollen or whatever it is, whatever that antigen is attached to. It will signal the phagocytic cell to come and engulf that whatever it is and bring it in and digest it. And then also antibodies can help with clumping various antigen containing molecules together so that it makes it easier for the immune system to see it. So kind of like we talked about before with complement cells are able to stick various microbes together. B cells can do the same thing. So let's say you're responding to some antigen on a grain of pollen. Your antibodies can help stick various pollen molecules together so that your phagocytic cells will recognize all of that kind of big glob of potentially harmful material and digest it. Now, of course, in the case of pollen, it's not actually dangerous to us, but let's say they were microbes, it can help accumulate those microbes together so they can all get phagocytosed. And so that's what they're showing you in this picture here. Antibodies can also play a role in preventing infectivity. So they can bind to various proteins that are on microbes. And when they're bound, it inhibits the microbes from being able to actually infect your cells. So it's almost like a physical block. And so that's what they're showing you here is that if that pathogen is coated in antibodies, it can't infect your cells, which helps keep you from getting sick and helps to prevent the pathogen from spreading. And then finally, antibodies can also signal complement proteins to come and break open different pathogens. So that's what they're showing in you this picture here. All these kind of like purple seed looking things in this image are the complement proteins, and they're being signaled by the antibodies to come poke holes in these pathogens so that they can help destroy some of those pathogens. Antibodies do not directly kill any pathogens. They just help with allowing the immune cells to identify those pathogens and complement cells, recruiting them to help destroy pathogens. But they don't do that themselves. The other type of adaptive immune cell are T cells. And T cells are also produced in the bone marrow, but they mature in the thymus. And just as a reminder, T cells are primarily responsible for cell-mediated immunity. So they're attacking cells, your cells, that have been infected by some sort of pathogen. So it can be a virus, or bacteria, or anything else like that. And so the T cells, they have several different types. And we'll go over 3 different types in this presentation. One, we have cytotoxic T cells, and these are our effector cells. They go around patrolling for your cells that have been infected with some sort of pathogen. And then once they encounter infected cells, they can do one of 2 things. They can poke holes in those cells. And as we talked about before, when you poke holes in cells, that causes water to rush in and those cells essentially burst and die. Or they can induce those cells to undergo apoptosis. And so apoptosis, just as a reminder, is programmed cell death. This is actually a relatively neat way of cell death. It's not like the cell explodes. Instead, the cell decides to start dying from the inside out. And so the T cell will bind to this infected cell and it'll start a series of events that'll cause the infected cell to start breaking down all the proteins and sugars and everything and DNA inside that cell. and then the cell will start to kind of be packaged into these vesicles, and then those vesicles will be consumed by phagocytic cells and degraded. And so that's what's being shown in this picture here. This cell is a cell that's undergoing apoptosis, and it looks kind of bubbly and bumpy like that because there's a whole bunch of different vesicles that will then be consumed by phagocytic cells. This is not what the cell normally looked like. This is what happens due to apoptosis. So this is a really effective way at kind of containing the pathogen while also killing the pathogen, because you don't rupture the cell and release whatever pathogen is to the cells around it. Instead, you just kind of fold that cell within itself and destroy it, and it's nice and clean and gone. And so these cytotoxic T cells generally reside in your lymph nodes and spleen, and they are activated and matured by helper T cells. And helper T cells are primarily responsible for stimulating and activating B and T cells. They do not actively fight infections. Instead, they produce cytokines that tell B cells and T cells to undergo clonal selection. And so we talked about this a little bit when we talked about that B cells can be antigen presenting cells. And I mentioned that B cell can show an antigen to a T cell. If that T cell is a helper T cell, then the helper T cell will say, oh, okay, you need to start making more of yourself so we can fight this infection. So it'll make cytokines that B cell will recognize, and the B cell's like, oh, I need to divide, and it'll undergo clonal selection. Similar thing happens with T cells, especially the cytotoxic T cells and the helper T cells. And so these helper T cells also generally reside within the lymph nodes and spleen. And then finally, we have our memory T cells. And memory T cells can be derived from either cytotoxic or helper T cells. And they function in a very similar way to the memory B cells, where they circulate throughout the lymphatic system just to make sure that they don't see that antigen again. So that if you do get reinfected and these memory T cells are circulating through your lymphatic system, they'll more quickly find that antigen and undergo clonal selection, and be able to quickly wipe out that infection because it's your second time being exposed. And so how this role between helper T cells and cytotoxic T cells works is kind of shown in this kind of graphic on the side here. So we start with an antigen presenting cell. And so this can be a dendritic cell, a macrophage, or a B cell will be presenting some sort of antigen on their cell surface. And then they'll travel to the lymph nodes. And in the lymph nodes, they take that antigen and bind and like interact with a lot of different helper T cells and say, hey, I see this antigen. Are you the T cell that fights it? No. Are you the T cell that fights it? No. Are you the T cell that fights it? No. And then until they keep doing this, until they find the helper T cell that has a receptor on its membrane that matches the antigen that antigen presenting cell is showing. And then once the helper T cell binds to that antigen, it will stimulate the production of cytokines. And the cytokines, which are shown here in this picture, will go on to activate the cytotoxic T cells and helper T cells that recognize that same antigen to undergo clonal selection. So we'll have cytotoxic T cells that recognize whatever that antigen is, and helper T cells that recognize whatever that antigen is, rapidly replicating. Those cytotoxic T cells will then be activated by cytokines produced by the helper T cells to go out and start killing these infected cells. And so that's how this whole process works. Now, without the helper T cells, B cells and cytotoxic T cells do not go on to actively fight infection. It's a two-part system. You don't want your immune system just going and doing whatever it wants. That can become problematic, which we'll talk about in the next couple slides. So in order to prevent that from happening, we have a two-part system. You can bind and recognize your antigen, but we also want to make sure that this is an appropriate response. So without those cytokines from helper T cells, cytotoxic T cells will not mature, and they won't actually go out and kill infected cells. And so this can be problematic because the infection can continue to spread. Similar thing with B cells. If these B cells are not stimulated or activated by the cytokines produced by helper T cells, then they won't actually go out and fight the infection in the bloodstream. It'll just basically be hanging out, waiting for that signal. So we need these helper T cells. They play an important role, even though they're not actively fighting the infection by killing the pathogen itself. Sometimes the immune system can go a little haywire. It gets a little confused, and it'll start to either attack your own body cells, or it can start to attack random stuff that's not even harmful to the body. And so when your immune system starts to attack your own body cells, this is called autoimmunity. And so this is a result of the fact that there are immune cells in your body that recognize these particular antigens. But those antigens are sugars or proteins or lipids that are naturally produced by your healthy body cells. And so what happens is the immune cells are just doing what they do. They recognize an antigen, they kill whatever thing that has that antigen. And so with that being your own cells, your immune system will attack your organs and tissues that have whatever cells that have that particular antigen. And so it's just basically they don't recognize self as self anymore. They see self as non-self. And autoimmune diseases can result in serious illness. They can also result in damage to tissues and organs, sometimes irreversible damage. And if it's bad enough, it can result in death. Autoimmune diseases can occur due to genetic mutations, environmental factors, and even some infections. So just another reason to try to stay as healthy as possible. Certain microorganisms out there can cause you to develop an autoimmune disorder. There are various autoimmune diseases out there. Some examples I provided here. Type 1 diabetes. We talked about type 1 diabetes in our last lecture on nutrition and digestion, and how when you have type 1 diabetes, your pancreas is not producing enough insulin, and so that's why you have a lot more sugar circulating in your blood. This is actually a form of autoimmune disorder, because what's happening is your own immune cells recognize proteins on your insulin producing cells of the pancreas and starts to attack them. So people don't make enough insulin who have type 1 diabetes, because every time your pancreas makes these insulin producing cells, your own immune system attacks those insulin producing cells and kills them. And so for these people, the only way that you can uptake the blood from your sugar from your blood is to either inject insulin or have an insulin pump. Then we also have multiple sclerosis, which is an attack on the insulation around the nerve fibers of your brain and spinal cord. And we'll talk about what that insulation is in our nervous system lecture. But this usually occurs in people between their 20s and 40s. You start to see some symptoms of this will arise. And then we have rheumatoid arthritis, which is an attack on the connective tissue in your joints. Graves' disease, which is an attack on the thyroid gland. Inflammatory bowel disease, which is an attack on the microbiota of your colon. So in that case, it's not necessarily recognizing your own cells as non-self cells, but it's recognizing the good guys that are helping you out in your colon as bad guys, which is not good either. And then we also have lupus. And lupus is an attack on various tissues in the body. So this can result in cell death and tissue and organ death at various parts around your body. And so in this chart here, I've included just because I think it's really good to show that there are various parts of the body that can be attacked. Sometimes this autoimmune diseases are localized to specific organs. So like we talked about with type 1 diabetes, only really attacking insulin producing cells of the pancreas, or Graves' disease attacking the thyroid gland, but some of them are a little bit more systematic, like lupus. And so this is just to show you that the list that I provided here is not all inclusive. There are a variety of autoimmune disorders out there. When your immune system mounts a strong immune response to pretty much harmless compounds and substances, this is referred to as allergies. And antigens that stimulate this response are called allergens. And there are a variety of allergens out there. I listed a few here that are really, really common: pollen, peanut butter, milk, eggs, shellfish, dander, and wheat. But there are hundreds, thousands of allergens out there. I once had a roommate who was allergic to a very specific protein made by caterpillars. Like almost anything can be an allergen as long as it's a innocuous substance that stimulates an inappropriate response from your immune system. And the symptoms that we associate with allergies are due to the histamine and cytokine release from mast cells. So we talked about this before when we talked about the innate immune system and how histamine and cytokines help with inflammation so that you can clear a microbial invasion. However, in this case, we don't necessarily want those histamines being produced because this is a harmless substance, but your immune system doesn't know that, right? So it responds as if this substance was harmful. And this response is antibody mediated. So what happens is the first time you're exposed to that allergen, your body starts to do a normal humoral immunity response. And so it makes a lot of antibodies. And then it'll clear whatever that allergen is, and those antibodies will hang around. And some of those antibodies go to mast cells. And I included a picture of a mast cell here. And they will go and bind to the mast cells, and they will just hang out there for a long period of time. And then what happens is the second time you get exposed to that allergen, those antibodies that are on those mast cells will activate and stimulate that mast cell to release all of the histamines that it has in its cytoplasm. So in this picture here, they're showing you all of the various vesicles with histamines. So all of these circles here are vesicles containing histamines. So what those mast cells will do is they will release all of those histamine vesicles and release the histamines into the environment. And that's what stimulates that strong immune response to something small like pollen or dander or I don't know, anything that you're potentially allergic to, right? There are so many different allergies out there. I'm sure most of you guys who are watching this have some sort of allergy. And so the allergic reaction can manifest itself as several different symptoms. I've listed a few here. You can get hives, which is like a rash on your skin. You have vomiting, diarrhea, hay fever, which is really, really common, especially if you live in the South because of all the pollen we get in the spring, asthma, and one of the most serious is anaphylactic shock. And this allergic reaction can be local or it can be systemic. So if you are like, I don't know, if you're allergic to milk, right? So let's say you're lactose intolerant. It might be a more local response where it's isolated to your colon and your small intestine and your gut, right? But it can be something that's more systematic, depending on what it is, especially if it gets into the bloodstream, then your whole body will react. And when that happens, you can have a greater chance of going into anaphylactic shock. So to combat allergies, we have a couple of different methods. If your allergy is not too severe, you can use antihistamines and steroids. And these will can do a couple of things. It can either block histamine and cytokine production. They can block the histamine and cytokine release from the cells. So the allergen will bind to those antibodies that are on the mast cells, but the mast cells won't release the histamines, so you don't get that overreaction. And then you can also have antihistamines and steroids that block the reception. So the histamines are being produced and they're being released into the environment, but the effector cells or the target cells do not see that the histamines are there, so they don't respond inappropriately. And then if your allergy is more severe and it could possibly lead to anaphylactic shock, then you may have to have an epinephrine pen or EpiPen. And this is a form of adrenaline that helps to combat anaphylactic shock. So during anaphylaxis, your throat will start to close up, you will have a difficulty breathing, and your blood vessels will constrict. And so this adrenaline, shot of adrenaline helps to keep your blood vessels from constricting so that you can actually breathe and counteract that shock experience. And if you ever have any questions about, do I have an allergy to anything out there, you can always get an allergy skin test. And this is a picture of what it looks like. I know it looks like it's crazy, but what happens, and I'm sure some of you guys have had this happen before where you've gone to get this test, they basically will inject various antigens underneath your skin, and then you just wait and see what happens. So in this case, this gentleman is very sensitive to a lot of antigens, because if your immune system reacts to that allergen, then you might see some sort of redness or welt or bump. So some examples of things that this particular individual is allergic to are like birch, right? Because you can see that here. Live oak is very allergic. Very allergic to almost all of these grasses, right? But he's not allergic to, let's say, what is this? Lamb's quarter, whatever that is. I don't know what that is, but he's not allergic to that, or sycamores, right? Because you don't see that allergic response. You don't see that immune response to those particular antigens. So this type of test is very effective, especially if you might have several different type of allergies, or you risk going into anaphylactic shock. This is a very effective way of figuring out what those allergens are for you, so that you can avoid them, so that you can avoid any sort of negative health consequences associated with exposure to those allergens. The last topic I would like to discuss in this lecture is AIDS, more specifically AIDS as it relates to the immune system. So we've already talked about HIV and AIDS in our microbiology lecture. So if you need a refresher on what HIV is, what AIDS is, how HIV infects cells, go back to that lecture to get a refresher. In this lecture, we'll just focus on HIV as it relates to the immune system. So HIV is human immunodeficiency virus, and it infects a couple different types of immune cells, but it really focuses on your helper T cells. It can infect macrophages as well, but helper T cells is its ideal host. And over time, infection with this virus can shut down your adaptive immune system. And this is due to a deficiency in helper T cells. So the virus itself kills some of your helper T cells, but also your cytotoxic T cells will kill infected helper T cells. So when you have both the virus and your immune system decreasing the helper T cell count lower and lower and lower, over time, you end up with such a low amount of helper T cells that your adaptive immune system can't even be stimulated. And this is what results as AIDS. And when you have AIDS, this leaves your body susceptible to relatively simple infections, things that normally, if you had a regular functioning immune system, your body would be able to easily clear. But when you have AIDS, your body can't fight back. So people end up dying from what would normally be a really simple infection to combat. So the shutdown of your immune system by HIV has a couple different stages. We begin with the acute phase. And in the acute phase is from the time you're infected to the time that you really stop seeing symptoms. And most people have flu-like symptoms during this time. A lot of people think they just have the flu or some other infection. They don't realize that they've been exposed to HIV. So what happens is the virus gets into your body and begins to infect your helper T cells. Well, while this is happening, it's stimulating both a humoral and cell-mediated immune response. So your B cells are making lots of antibodies to bind to the virus so you can remove it, as well as trying to identify the virus in the bloodstream to intake it and break it down. And then your cell-mediated response, your cytotoxic T cells are patrolling the body, looking for helper T cells that are infected with the virus and killing those. So once this process is over, it usually kills the vast majority of infected helper T cells and virus. And over time, the antibodies will also decrease in the blood, but the high titers or the high amounts of anti-HIV antibodies that circulate in the bloodstream will be high for a relatively long period of time, like several months. And this is how, when you think that you've been infected with the virus and you go and get tested, they will do an antibody test to see if you have those antibodies against HIV in your blood. And if so, then they'll be able to tell you you're positive, and if not, you're negative. Now, if you wait too long, you have to use different types of testing. and we'll talk about why in a second. So before we move on, I want to go down to this chart down here. It's a really good chart. We've always used it when I was taking my virology classes to explain kind of what's happening between the T cells in your body and the virus in your body. So when we start from infection here, we just follow the virus over the first couple of days. So up to, well, yeah, days and then months, right? So in the first couple of days when you get the virus, the number of viral particles is increasing rapidly, like insane, right? They're replicating. That's because, like we talked about before, the adaptive immune system takes a little while to catch up to a pathogen. So during that time, the virus is going from helper T cell to helper T cell, just spreading, killing cells, spreading, killing cells. And so the viral numbers increase until, let's follow with green, your immune cells, your T helper cells, start to recognize the virus. So you can see as the viral titer increases, the number of helper T cells decreases. And that's because the virus is going into those helper T cells and killing those cells. But then eventually the cytotoxic T cells kick in, your B cells kick in, and some of the healthy helper T cells kick in and start to recognize the infection. And so your number of viral particles will start to decrease, right? Because you're effectively removing the vast majority of virally infected cells and viral particles. And this will allow for your number of helper T cells to increase. And this is because new helper T cells will be produced to replace those ones that you lost in the battle against the HIV virus. But the story doesn't end here. What happens is there are some viral particles that are basically hiding in your helper T cells, and they'll be dormant there for a period of time. It could be weeks, it could be months, but they're just kind of hiding there. And then some of those viral particles aren't really hiding so much. They're being infective. So they're infecting helper T cells, moving to the next cell, infecting helper T cells, moving to the next cell. But at this point, you don't see any symptoms. And it's because it's not a huge response like we saw before. The HIV virus is now infecting helper T cells and hiding, and then infecting other helper T cells and hiding. And as this process is going on, your helper T cells are being killed by the virus, as well as your immune system occasionally sees those infected helper T cells. They're trying to kill those cells off. But this is a slow process. And so what happens during this time to your viral numbers is they seem pretty low, right? They're pretty low numbers for months, years even, right? And everything seems fine. You don't feel sick. Everything is okay. But since your body is still fighting that infection, the number of helper T cells will slowly start to decrease as the number of viral particles is increasing. And then eventually you end up with so few number of helper T cells due to the virus either killing them off or your cytotoxic T cells killing them off that you can't even fight off any other infections. And this is where we enter into the last phase, which is the phase of AIDS. And this is where your the body is no longer able to fight off basic small infections, and you can end up dying from something as small as a common cold or thrush, that is a yeast that will form in your mouth. So this is a very serious illness. There are a lot of medications out there. You've probably seen them on TV advertised that are helping to combat this virus, but there's still no cure. So you want to make sure that you're protecting yourself, wearing condoms. If you are in a clean needle exchange program, support those programs, even if you're not a drug user, because it really does help people to protect themselves from this virus and various other illnesses and viruses and bacteria out there. Like there's a lot of risk when you're an intravenous drug user, as well as women who want to breastfeed, but they have our HIV. This can also lead to transmission from mother to child. So these people also need support until we can find a cure. Hopefully we can find a cure sometime soon. But at this point, there is no cure. So we have to keep working on finding effective ways to combat this virus and combat the spread of this virus. This concludes our lecture on the immune system. You can go ahead and read chapter 27, sections 1 through 8, and then 10 through 13, and also do the chapter 27 learning curve assignment. Then I've included here a variety of videos to help explain further provide clarity to the topics that we discussed in this lecture. And then some of them are just to visualize some of the things that we talked about. They're either animations or actual video of, like for example, the one here that says immune cell eating bacteria is an actual video of a real neutrophil eating bacteria. And the bacteria have been stained green so you can see them. So that video is kind of cool. But as you're going through these videos, some of them are a little bit redundant, but they have differences in their complexity and depth of the topic. So some of them are a little bit more surface level, like the how does the immune system work and the immune system explained is a little bit more surface level, whereas the crash course videos are a little bit more in-depth on each of the topics. So you might want to start with the surface level videos, then go to the videos that are more in-depth. And then some of the videos also have details that are beyond the scope of this course. So just keep that in mind as you're watching them. A good example of this is the video that says types of immune cells. I would only watch that video to about 3 minutes and 53 seconds, because after that, she starts to talk about the specific immune cell receptors and what they do and their names. You don't need to know that much depth in the class. So just remember the things that we talked about in the lecture are key for you to understand, and then all the kind of more minor details, you don't have to memorize those at this point, but they may help you to connect the dots between the various concepts that we've talked about in this lecture. So with that said, this is, we only have one more lecture. This is our second to the last, or we might have 2 more lectures depending on if time permits. So we're almost done, and next we'll talk about the nervous system. So I'll see you next time.